So here's something worth knowing as you settle in tonight. Right now, inside your body, there are roughly 37 trillion cells. That's 37 followed by 12 zeros. And tucked inside almost every single one of those cells, coiled up and packed away with extraordinary precision, is a molecule so long that if you stretched out just one copy of it, it would reach about 6 feet. Taller than most people, and you have a copy in nearly every cell. If you took all the DNA in your body and laid it end to end, it would stretch to the sun and back more than 300 times. That's not a metaphor, that's arithmetic. And all of that DNA, every last nucleotide, is carrying instructions. Instructions for building you, for keeping you alive, for repairing the tiny damages that accumulate every day, for the color of your eyes and the shape of your earlobes, and the way your immune system recognizes a virus it's never seen before. It's the most information-dense storage system known to exist anywhere in the universe, and it's been running, essentially without interruption, for about 4 billion years. And that's what we're going to talk about tonight. DNA and genetics. The whole story, from the very beginning, from a time before anyone even knew that genes existed, all the way through to the technologies that are reshaping medicine and agriculture and criminal justice, right now, tonight, as you lie here. We're going to take it slow. We're going to meet the people who figured this out, the ones who got it right, and the ones who got it spectacularly wrong. The rivalries and the accidents, and the moments where everything changed because someone noticed something that everyone else had overlooked. And if you drift off somewhere along the way, that's perfectly fine. The story will be here. Honestly, some of this stuff is so beautifully strange that it almost makes more sense when your mind is loose and unhurried, not trying too hard to pin everything down. Let's start where the story really starts, which is with a question that humans have been asking for as long as there have been humans. Why do children look like their parents? Think about how obvious and yet how deeply mysterious that is. A baby is born, and people gather around and say, "Oh, she has her father's nose, she has her mother's eyes." Farmers have known for thousands of years that if you breed the biggest sheep together, you tend to get bigger lambs. If you plant seeds from the sweetest melons, you tend to get sweeter melons. Inheritance is one of the most visible facts of life, and yet, for most of human history, nobody had the faintest idea how it actually worked. What was being passed from parent to child? How? Through what mechanism? The theories people came up with were, honestly, all over the map. Hippocrates, around 400 BCE, thought that every organ in the body produced tiny particles, little seeds, that collected in the reproductive fluids and carried the characteristics of each body part. Aristotle disagreed. He thought the father contributed the form, the plan, and the mother contributed the raw material. For centuries, people debated variants of these ideas without making much progress. And then, in the mid-1800s, in a monastery garden in what is now the Czech Republic, a quiet, meticulous monk started counting peas. His name was Gregor Johann Mendel, and if there's a single person who deserves to be called the founder of genetics, it's him. But here's the thing about Mendel. He was not famous in his lifetime. He published his results in 1866, and almost nobody noticed. Almost nobody read the paper. Almost nobody understood what he'd done. He died in 1884, without any recognition for what would turn out to be one of the most important scientific discoveries of the 19th century. And the story of how that happened, how this man cracked the fundamental rules of inheritance, and the world just shrugged, that's worth telling properly. Mendel was born in 1822, in a small farming village in Silesia, then part of the Austrian Empire. His family were farmers, not wealthy. Young Mendel showed intellectual promise early, but money was a constant problem. His younger sister actually gave up part of her dowry so he could continue his education, which tells you something about the family's values and also their financial situation. Eventually, at 21, he entered the Augustinian Abbey of St. Thomas in Brunn, which is now Brno. And this wasn't primarily a spiritual decision, although Mendel was genuinely devout. The monastery was one of the few places where a young man without money could get access to education, to books, to scientific equipment, to colleagues who cared about ideas. The abbot at the time, Cyril Knapp, was himself interested in breeding experiments and had been encouraging the monks to study heredity in plants and animals. So Mendel found himself in exactly the right place. He tried to become a teacher and failed the certification exam, twice actually. The second time, in 1856, he apparently had some kind of breakdown during the exam and couldn't finish it, so he never became a certified teacher, which is one of those ironies that history seems to enjoy. Instead, he went back to the monastery and started his experiments. And what he did was, on the surface, incredibly simple. He grew pea plants. But the way he grew them was anything but simple. Mendel chose garden peas, Pisum sativum, for very specific reasons. They were easy to grow. They grew quickly. They had clearly distinct either-or traits. The peas were either smooth or wrinkled, either yellow or green. The flowers were either purple or white. The plants were either tall or short. There was no ambiguity, no blending, no middle ground. And critically, pea plants can self-pollinate, which means you can control exactly which plant breeds with which. Mendel could take pollen from one plant and carefully transfer it to another, then cover the flower to prevent any stray pollen from interfering. He was an obsessive record keeper. Over the course of 8 years, from 1856 to 1863, he cultivated and studied something like 29,000 pea plants. Think about the patience that requires, the daily attention, the meticulous cataloging of every cross, every offspring, every characteristic. And here's what he found, and this is beautiful. When he crossed a pure-breeding tall plant with a pure-breeding short plant, all the offspring in the first generation were tall. Not medium, not a blend, tall, every single one. The shortness seemed to have vanished entirely. But then, when he let those first-generation tall plants self-pollinate, something remarkable happened in the second generation. About 3/4 of the plants were tall, and about 1/4 were short. The shortness was back. It had been hiding, somehow, in the first generation, invisible but still there, and now it reappeared in a precise, predictable ratio, 3:1, tall to short. And he found the same pattern for every trait he studied: smooth versus wrinkled seeds, yellow versus green, purple flowers versus white. Always that same pattern. One trait dominated in the first generation, the other vanished, and then the hidden trait re-emerged in the next generation in a 3:1 ratio. Now, you might be thinking, okay, 3:1, that's an interesting number. But why does it matter? And here's where Mendel's real genius shows. He didn't just observe the ratio, he explained it. He proposed that each trait is controlled by what he called factors. We'd call them genes today. And that each organism carries 2 copies of each factor, one inherited from each parent. Some factors are dominant, meaning they mask the presence of the other. And some are recessive, meaning they only show their effect when both copies are the recessive type. When a plant has one dominant and one recessive factor, it looks just like a plant with 2 dominant factors. The dominant one wins. But the recessive factor is still there, still being carried, still capable of being passed on to the next generation. Think about what this means. Mendel had essentially figured out that inheritance is particulate, not blending. The traits don't mix like paint. They're carried by discrete units that maintain their identity across generations. They can be shuffled and recombined, but they don't dilute or disappear. A short plant crossed with a tall plant doesn't produce medium plants. It produces plants that carry both instructions, one for tall and one for short, and the tall instruction happens to be the one that gets expressed. But the short instruction is still there, intact, ready to emerge in future generations if the right combination occurs. This was revolutionary. It was the foundation of everything that would follow in genetics, and the scientific community almost completely ignored it. Mendel presented his findings to the Natural History Society of Brünn in 1865, and they were published in the Society's Journal in 1866. The paper was sent to about 40 libraries and scientific institutions across Europe. A few people read it. One prominent botanist, Carl von Nageli, actually corresponded with Mendel about it, but didn't really understand the significance. Nageli suggested that Mendel repeat his experiments with hawkweed, a plant that, as it turned out, reproduces in unusual ways that made it terrible for genetic studies. Mendel spent years struggling with hawkweed and couldn't replicate his pea results, which must have been deeply frustrating. Eventually, in 1868, he was elected abbot of the monastery, and administrative duties consumed most of his time. He grew heavy, developed kidney problems, and died in 1884 at the age of 61. His papers were burned after his death by the next abbot, who was worried about a tax dispute with the government and wanted to destroy any personal documents. And so, the foundation of genetics lay dormant for 34 years. 34 years. That's extraordinary when you think about it. One of the most important insights in the history of biology, published in a legitimate scientific journal, accessible to anyone who cared to read it, and the world just moved on without noticing. It's a reminder that great ideas don't always arrive with trumpets. Sometimes they arrive quietly, in a monastery journal, and wait patiently for the world to catch up. The catching up happened in 1900, and it happened almost simultaneously in 3 different countries, which is one of those coincidences that makes you think the idea's time had simply come. 3 botanists, working independently, Hugo de Vries in the Netherlands, Karl Correns in Germany, and Erich von Tschermak in Austria, each rediscovered Mendel's principles through their own experiments. And each of them, in the process of searching the literature to see if anyone had done similar work, stumbled across Mendel's 1866 paper and realized, with varying degrees of grace, that a monk in Brünn had beaten them to it by more than 3 decades. De Vries initially tried to claim some of the credit for himself, but Correns made sure Mendel got his due. The paper was reread, re-evaluated, and suddenly recognized as the masterpiece it was. Mendel went from forgotten monk to the father of genetics, posthumously, overnight. And now, right at the turn of the 20th century, genetics was suddenly a real field, with real principles, and a rush of questions that needed answers. Mendel had shown that traits are carried by discrete factors that follow predictable rules. But what were those factors, physically? Where in the cell did they live? What were they made of? How did they copy themselves? How did they produce their effects? Mendel had given the world a set of laws without any mechanism. It was as if someone had discovered the rules of chess without ever seeing a chessboard or the pieces. The rules were brilliant, but the underlying reality was still completely mysterious. And the search for that underlying reality would take another 50 years. It would involve microscopes and radioactive isotopes and X-ray photographs and one of the most famous and most controversial scientific races of the 20th century. It would end with the discovery of a molecule so elegant, so simple in its architecture, and yet so breathtakingly powerful in its function, that when the scientists who found it first saw its shape, one of them reportedly walked into a pub in Cambridge, England, and announced that they had discovered the secret of life. But we're getting ahead of ourselves, because between Mendel's rediscovery in 1900 and that moment in the pub in 1953, a tremendous amount had to happen. The first thing was that scientists needed to figure out where in the cell these mysterious Mendelian factors actually lived. And the answer to that question was already starting to emerge, thanks to improvements in microscope technology and some very careful observations of cell division. Because when you look at cells dividing under a microscope, you can see these strange, dark-staining, thread-like structures that appear and then split apart and move to opposite ends of the cell just before it divides in two. Those thread-like structures were first described in detail in the 1880s by a German biologist named Walter Flemming, who stained cells with aniline dyes and watched the process of cell division unfold under his microscope. He called the material that absorbed the dye chromatin, from the Greek word for color. The structures formed from chromatin during cell division would eventually be called chromosomes, meaning colored bodies, and Flemming carefully documented how, during cell division, the chromosomes duplicated themselves and then separated, so that each daughter cell received a complete set. He called this process mitosis, and his drawings from the 1880s are still remarkably accurate. Now, here's where things start to connect. By the early 1900s, several scientists were looking at chromosomes and thinking about Mendel's factors, and a light was starting to come on. The American biologist Walter Sutton and the German biologist Theodore Boveri, working independently around 1902 and 1903, both proposed what became known as the chromosome theory of inheritance. The idea was simple and beautiful. Mendel's factors, whatever they were, must be located on chromosomes. Chromosomes come in pairs, just as Mendel's factors do. During the formation of sex cells, sperm and egg, the chromosome pairs separate, so that each sex cell gets only one chromosome from each pair. Just as Mendel said, each gamete carries only one copy of each factor. When sperm meets egg, the pairs are restored. The parallel was too perfect to be coincidence, but not everyone was convinced. Correlation is not causation, as they say, and at this point, the chromosome theory was still circumstantial. The evidence was suggestive, but not definitive. And the person who would turn that circumstantial case into an airtight one was a skeptic himself, a man who started out doubting the chromosome theory and ended up providing its most compelling proof. His name was Thomas Hunt Morgan, and his story is one of the great detective tales in the history of biology. Morgan was a Kentuckian, born in Lexington in 1866, the same year Mendel published his paper. He was a zoologist at Columbia University in New York, and in the early 1900s, he was frankly unimpressed by both Mendelian genetics and the chromosome theory. He thought the evidence was too thin, the reasoning too speculative. He was an experimentalist, a man who wanted to see things with his own eyes, and what he saw when he looked at the Mendelian framework was a theory that was long on logic and short on direct physical evidence. So he set out to test it, and the organism he chose to test it with was a tiny, humble creature that would become one of the most important animals in the history of science. Drosophila melanogaster, the common fruit fly. You've seen them. Those little flies that hover around overripe bananas on your kitchen counter. Morgan chose them for the same kind of practical reasons that Mendel chose peas. Fruit flies are cheap to maintain. You can keep thousands of them in a single milk bottle. They breed incredibly fast, a new generation every 2 weeks. They have only 4 pairs of chromosomes, which makes the genetics simpler to track. And they produce hundreds of offspring at a time, which means you can spot rare mutations and calculate ratios with statistical confidence. Morgan's lab at Columbia became famous. It was called the Fly Room, and it was exactly as unglamorous as it sounds. A small, cramped room, about 16 by 23 feet, filled with milk bottles full of rotting bananas and swarming with fruit flies. The smell, by all accounts, was spectacular. Morgan and his students worked there for years, breeding flies by the millions, inspecting them under magnifying glasses, recording every tiny variation in eye color, wing shape, body pigment. They were looking for mutations, for anything unusual, for deviations from the norm that they could track through generations and map to chromosomes. And then, in 1910, Morgan found what he was looking for. Among the thousands of red-eyed flies in his bottles, he spotted a single male fly with white eyes. Just one, a natural mutation, and Morgan, the careful experimentalist, immediately started breeding it. He crossed the white-eyed male with a normal red-eyed female, and in the first generation, all the offspring had red eyes. Classic Mendelian dominance. Red was dominant over white. So far, so expected. But then he let the first generation interbreed, and in the second generation, the white eyes reappeared, roughly 1 in 4 flies, just as Mendel would have predicted, except for one crucial detail. All the white-eyed flies were male. Every single one. Think about that for a second. Why would eye color have anything to do with sex? In Mendel's framework, traits are supposed to sort independently of each other. But here was a trait, eye color, that was linked to sex. Morgan realized what was going on. The gene for eye color must be physically located on the sex chromosome. In fruit flies, as in humans, sex is determined by a pair of chromosomes. Females have 2 X chromosomes. Males have 1 X and 1 Y. If the white eye gene was on the X chromosome, then the inheritance pattern made perfect sense. A female fly could carry the white eye gene on 1 X chromosome, but still have red eyes, because the other X chromosome carried the dominant red eye gene. But a male fly only has 1 X chromosome. If that 1 X carries the white eye gene, there's no second X to mask it. The white eyes show up. This was the smoking gun. Morgan had shown that a specific gene was on a specific chromosome, not just correlated with chromosomes in a vague, hand-wavy way, but physically located on a particular chromosome that you could see under a microscope. Mendel's abstract factors had a physical address, and Morgan, the skeptic, became the champion of the chromosome theory. He spent the next 2 decades, together with a brilliant group of students, Alfred Sturtevant, Calvin Bridges, Hermann Muller, mapping more and more genes to specific locations on fruit fly chromosomes. Sturtevant, when he was still an undergraduate, pulled an all-nighter and created the first genetic map, figuring out the order of genes along a chromosome based on how frequently they were inherited together. The closer 2 genes are on a chromosome, the less likely they are to be separated during the shuffling that happens when sex cells form. So by tracking how often traits were co-inherited, you could figure out how far apart the genes were. It was brilliant, and Sturtevant did it in 1 night, at the age of 19. Morgan won the Nobel Prize in 1933 for this work, and the fruit fly became, and remains to this day, one of the most studied organisms on Earth. Thousands of laboratories around the world still work with Drosophila, and the genetics of this little fly has taught us an astonishing amount about how genes work. not just in flies, but in humans, because the fundamental mechanisms are shared across almost all living things. The genes that control how a fruit fly's body plan develops, which end is the head, where the wings go, where the legs attach, have direct counterparts in human DNA. We are far more closely related to flies than you might want to think about as you're lying here trying to fall asleep. But here's the thing. Morgan and his students had shown where genes live, on chromosomes. They'd mapped the relative positions of genes along those chromosomes. They'd demonstrated the physical basis of Mendel's laws. But they still didn't know what genes were made of. What was the actual substance? What was the chemical nature of the gene? Chromosomes are made of two types of molecules, proteins and nucleic acids. And for decades, almost everyone assumed that genes must be made of protein. The logic seemed obvious. Proteins are enormously complex molecules, made of 20 different amino acids that can be arranged in virtually infinite combinations. They have the structural diversity, the chemical versatility, to encode all the varied information that a gene would need to carry. Nucleic acids, on the other hand, DNA, specifically, were thought to be boring, chemically monotonous. A simple, repetitive molecule made of only 4 building blocks, probably just structural scaffolding, the molecular equivalent of packing peanuts, holding the important stuff in place. This was called the tetranucleotide hypothesis, the idea that DNA was just a boring, repeating sequence of its 4 components. It couldn't possibly carry genetic information. It wasn't complex enough. That assumption was wrong, deeply, consequentially wrong, and overturning it would take decades and some of the most elegant experiments in the history of biology. The first crack in the assumption came from an unexpected direction, a British medical officer studying pneumonia in the 1920s. His name was Frederick Griffith, and in 1928, he performed an experiment that he didn't fully understand at the time, but that would change the course of biology forever. Griffith was working with Streptococcus pneumoniae, the bacterium that causes pneumonia. There were 2 strains. The virulent strain, which caused disease, had a smooth polysaccharide capsule around each cell, so it was called the S strain for smooth. The harmless strain lacked this capsule and formed rough-looking colonies, the R strain for rough. Simple enough. S kills mice, R doesn't. Here's what Griffith did. First, he confirmed the basics. Inject mice with live S bacteria, the mice die. Inject mice with live R bacteria, the mice live. Inject mice with heat-killed S bacteria, bacteria that have been cooked, destroyed, thoroughly dead. The mice live. No surprises so far. But then he tried something that nobody expected to matter. He mixed heat-killed S bacteria with live R bacteria and injected the combination. The heat-killed S cells were dead. The live R cells were harmless. Logically, nothing bad should happen. The mice died. And when Griffith examined the dead mice, he found live S bacteria in their blood. Smooth-capsuled, virulent, very much alive S bacteria. But the only live bacteria he'd injected were the harmless R strain, and the only S bacteria he'd used were thoroughly dead. Something from the dead S bacteria had gotten into the live R bacteria and transformed them, changed them, permanently. The harmless R bacteria had been converted into deadly S bacteria, and the transformation was heritable. The newly virulent bacteria passed the trait to their offspring. Griffith called this the transforming principle. Something, some chemical substance, had passed from the dead cells to the living ones and permanently altered them. It was as if a dead organism had reached out and rewritten the genetic instructions of a living one. Griffith didn't know what the substance was. He published his results, and they created a stir. But the identity of the transforming principle remained a mystery for another 16 years. And this is where the story takes one of those turns that makes you appreciate just how much patience and persistence real science requires. Because the person who identified the transforming principle, who proved what it was made of, was a quiet, meticulous, somewhat underappreciated scientist at the Rockefeller Institute in New York. His name was Oswald Avery, and he spent the better part of a decade working on this problem, enduring skepticism and doubt before publishing the answer in 1944. And the answer shocked nearly everyone. The transforming principle was DNA, not protein, DNA. Avery, together with his colleagues Colin MacLeod and Macklin McCarty, performed a beautifully systematic experiment. They took the extract from dead S bacteria, the stuff that could transform R bacteria into S, and methodically destroyed different components of it. When they destroyed the proteins, the extract still worked. It could still transform bacteria. When they destroyed the lipids, it still worked. When they destroyed the RNA, it still worked. But when they treated the extract with an enzyme that specifically destroys DNA, an enzyme called DNase, the transforming ability vanished. It was DNA. It had to be DNA. The genetic material, the stuff that carried heritable information from one cell to another, was deoxyribonucleic acid. Now, you might expect that this would have been headline news, that the scientific community would have immediately embraced this finding and pivoted its entire understanding of genetics. But that's not what happened. The paper was received with skepticism, even hostility in some quarters. Many prominent scientists simply didn't believe it. The prejudice against DNA as a boring, structurally monotonous molecule was so deeply entrenched that people looked for ways to dismiss Avery's results. Maybe the DNA preparation was contaminated with trace amounts of protein, and it was really the protein doing the work. Maybe the experiment wasn't clean enough. Avery was a cautious man, not given to grand claims, and he knew the implications of his findings were enormous. He wrote in a letter to his brother in 1943 that if the results held up, they meant that DNA could constitute a transformation of the gene, but he hedged, as careful scientists do, noting that the evidence could still be interpreted in other ways. Avery never received the Nobel Prize, which is widely regarded as one of the great oversights in the history of the award. He died in 1955, just 2 years after the structure of DNA was finally revealed, without ever getting the recognition he deserved. But his 1944 paper was the turning point. Even if not everyone believed it immediately, it forced people to take DNA seriously, to look at this supposedly boring molecule with new eyes, to ask the question that would drive the next decade of research. If DNA really is the genetic material, then what does it look like, and how does it work? And it was that question, that urgent, tantalizing question, that would launch one of the most famous and most bitterly contested races in the history of science. A race that would involve a brash young American biologist, a loquacious English physicist turned biologist, a brilliant and undervalued English chemist, and a New Zealand-born physicist who had already won 1 Nobel Prize and was about to contribute to a discovery that would justify another. The prize was the structure of DNA, the three-dimensional shape of the molecule that carries the instructions for life. And the story of how that structure was found is a story of ambition, rivalry, sexism, stolen data, flashes of insight, and one of the most beautiful molecules anyone has ever seen. Let's talk about it. By the early 1950s, the evidence that DNA was the genetic material had become much harder to ignore. In 1952, Alfred Hershey and Martha Chase, at Cold Spring Harbor Laboratory, performed a famous experiment using bacteriophages, viruses that infect bacteria. These viruses are beautifully simple, just a protein shell surrounding a core of DNA. When a phage infects a bacterium, it attaches to the cell surface and injects something inside that hijacks the cell's machinery and forces it to make new copies of the virus. But what gets injected? The protein or the DNA? Hershey and Chase were clever about this. They grew 2 batches of phages. One batch was grown in a medium containing radioactive sulfur, which gets incorporated into proteins but not DNA. The other batch was grown with radioactive phosphorus, which gets incorporated into DNA but not proteins. So they had phages with labeled protein coats and phages with labeled DNA cores. They let each batch infect bacteria, then used a kitchen blender, literally, a Waring blender, the kind you might use to make a milkshake, to shake the empty phage shells off the outside of the bacteria, and then they separated the shells from the bacteria using a centrifuge. The result was clear. The radioactive phosphorus, the labeled DNA, was inside the bacteria, running the show. The radioactive sulfur, the labeled protein, was in the discarded shells on the outside. It was DNA, not protein, that entered the cell and carried the genetic instructions. Hershey and Chase's experiment, combined with Avery's earlier work, essentially closed the case. DNA was the stuff of genes, and now the race was on to figure out its structure. Because knowing that DNA is the genetic material tells you nothing about how it works. To understand the mechanism, how DNA stores information, how it copies itself, how it directs the construction of proteins, you need to know the shape, the three-dimensional architecture. And in the early 1950s, several groups around the world were gunning for that prize. Let's set the stage. There were 3 main players working in 2 cities in England. In London, at King's College, there was Maurice Wilkins and Rosalind Franklin. Wilkins was a New Zealand-born physicist who had worked on the Manhattan Project during the war. He'd been at Berkeley, working on isotope separation for the atomic bomb, and who had turned to biophysics afterward, partly out of a desire to do something more life-affirming with his skills. He had started doing X-ray crystallography work on DNA, and was getting some promising, if fuzzy, images. Then, in 1951, Rosalind Franklin arrived at King's College to lead her own X-ray diffraction work on DNA fibers. Franklin is one of the most important and most tragically treated figures in this story, and she deserves a proper introduction. She was born in London in 1920 to a prominent Anglo-Jewish family. She was fiercely intelligent, sharp, meticulous, and not inclined to suffer fools. She had earned her PhD at Cambridge for work on the microstructure of coal and other carbons, work that had direct practical applications in gas mask filters during the war, and then spent several years in Paris at the Laboratoire Central des Services Chimiques, where she became expert in X-ray crystallography. She was, by any measure, one of the finest experimentalists in the world at this particular technique. And here's where things get complicated, and frankly, a bit ugly. When Franklin arrived at King's College, there was a misunderstanding about her role. She believed she had been hired to lead an independent research program on DNA. Wilkins believed she had been brought in as his assistant. The misunderstanding was never properly resolved, and the two of them developed a terrible working relationship. Part of this was personality. Wilkins was quiet and diffident. Franklin was direct and forceful. Part of it was almost certainly sexism. King's College in the 1950s was not a welcoming place for women scientists. The senior common room, where researchers gathered for lunch and collegial conversation, was men only. Franklin had to eat elsewhere. She was isolated, socially and professionally. in a way that her male colleagues simply weren't. But she did extraordinary work despite all of this. Using painstaking experimental technique, she obtained X-ray diffraction images of DNA that were far sharper and more informative than anything anyone had produced before. She figured out that DNA existed in two forms depending on humidity. She called them the A form and the B form, and she focused her work on the A form, which gave more complex, but potentially more information-rich patterns. Meanwhile, she had also taken an image of the B form that would turn out to be one of the most consequential photographs in the history of science. Now, 60 miles away, in Cambridge, at the Cavendish Laboratory, there were James Watson and Francis Crick. Watson was an American, young. He was only 23 when he arrived in Cambridge in 1951, ambitious, brash, and obsessed with finding the structure of DNA. He had been trained in genetics, not X-ray crystallography, and he had read Erwin Schrödinger's famous little book, What is Life?, which argued that the gene must be some kind of aperiodic crystal, a molecule with a complex, non-repeating structure that could carry information like a code. That idea had captivated Watson and drawn him to the structural question. Crick was 12 years older, English, and hadn't even finished his PhD yet. He'd started in physics, had his thesis work interrupted by the war. He'd designed naval mines for the Admiralty, which is about as far from DNA as you can get, and had come to Cambridge to start over in biology. Crick was brilliant, garrulous, and had a laugh so loud and distinctive that it reportedly annoyed the head of the Cavendish Laboratory, Lawrence Bragg. But he also had an extraordinary ability to see through to the essential structure of a problem, to combine information from different sources and see what it meant. Watson and Crick were not doing experiments. This is an important point. They were not growing DNA crystals. They were not firing X-rays. They were building models, physically constructing models of what the DNA molecule might look like, using metal rods and clamps, trying to find an arrangement of atoms that was consistent with all the available evidence, the X-ray data, the chemical composition, the known bond angles and distances. It was a theoretical approach, almost playful, and it drew heavily on the method that Linus Pauling had used so brilliantly when he discovered the alpha helix structure of proteins in 1951. Pauling, based at Caltech in Pasadena, was the other major competitor in the DNA race, and Watson and Crick lived in terror that he would beat them to the answer. There's a famous story about an early, embarrassing failure. In late 1951, Watson attended a seminar by Rosalind Franklin in London, where she presented some of her X-ray data. He didn't take notes. He tried to remember the numbers she'd presented and got some of them wrong. He went back to Cambridge, and he and Crick built a model based on those faulty numbers. A triple helix with the phosphate backbone on the inside and the bases sticking outward. They invited Wilkins and Franklin up from London to see it, and Franklin immediately spotted the errors. She politely demolished the model. The water content was wrong. The structure was chemically impossible, as Watson and Crick had built it. It was embarrassing, and Lawrence Bragg, already irritated by the whole enterprise, told Watson and Crick to stop working on DNA and focus on their actual assigned projects. They didn't stop, of course, not really. They kept thinking about it, kept talking about it, kept waiting for the crucial piece of evidence that would unlock the puzzle. And in early 1953, two things happened that broke the case open. The first was Erwin Chargaff. Now, Chargaff was an Austrian-born biochemist at Columbia University who had been studying the chemical composition of DNA for years, and he had discovered something strange and beautiful. In any sample of DNA, from any organism, the amount of adenine, one of the four bases, always equaled the amount of thymine. and the amount of guanine always equaled the amount of cytosine. Always. A equals T, G equals C. These became known as Chargaff's rules, and they were a huge clue. They practically screamed that adenine pairs with thymine and guanine pairs with cytosine. But Chargaff himself didn't make the structural leap. He met Watson and Crick in 1952 and was, by most accounts, thoroughly unimpressed. He found Watson too young and ignorant, and Crick too talkative. The meeting was socially awkward, but the data Chargaff had published was right there, waiting to be used. The second thing that happened, and this is the part of the story that has generated the most controversy, was that Watson saw Franklin's X-ray photograph of the B form of DNA, the photograph known as Photo 51. How Watson got access to this image is a matter of considerable historical debate and ethical complexity. What is clear is that in January 1953, Maurice Wilkins showed Watson Photo 51 without Franklin's knowledge or permission. Wilkins walked into his office, pulled the photograph from a drawer, and showed it to Watson. Watson later wrote, with remarkable candor, that the moment he saw it, his mouth fell open and his pulse began to race. The photograph was an X-shaped pattern, a distinctive cross of dark spots, that is the telltale signature of a helical structure. And the specific pattern told you the dimensions of the helix, the width, the spacing between turns, the number of chains. Franklin herself had been cautious about interpreting her data. She was a careful, rigorous scientist, and she didn't want to jump to conclusions. She had been systematically working through the analysis of the A form, which was more complex. She had, in her own lab notebooks, already deduced many of the key features of DNA's structure, the helical nature, the sugar-phosphate backbone on the outside, but she was working toward her conclusions methodically, building the case one piece of evidence at a time. Watson and Crick worked differently. They took the information from Photo 51, which Watson remembered and Crick translated into structural constraints, and combined it with Chargaff's base pairing rules and with their model-building approach. And within a few weeks, everything fell into place. The structure of DNA is a double helix. Two long chains, wound around each other in a spiral, like a twisted ladder. The sides of the ladder are made of alternating sugar and phosphate groups, the sugar-phosphate backbone. And the rungs of the ladder are made of pairs of bases, adenine paired with thymine, guanine paired with cytosine, held together by hydrogen bonds. The two strands run in opposite directions, anti-parallel, which matters for how the molecule is copied, and the whole thing has a certain width, a certain spacing between base pairs, a certain twist per turn, all of which matched the X-ray data beautifully. But here's the thing that made Watson and Crick's discovery not just beautiful, but profound. The structure immediately suggested the mechanism of replication. Look at the base pairing. Adenine always pairs with thymine. Guanine always pairs with cytosine. Each strand of the double helix is a perfect template for the other. If you unwind the two strands and separate them, each individual strand contains all the information needed to reconstruct its partner. An A on one strand tells you there must be a T on the new strand. A G tells you there must be a C. You just read along the template and build the complementary strand, and you end up with two identical copies of the original double helix. Watson and Crick, in their famously understated 1953 paper in Nature, just over 900 words, one of the shortest landmark papers in scientific history, noted, with truly British restraint, that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. That might be the most famous understatement in the history of science. The paper was published on April 25, 1953. Watson was 25 years old. Crick was 36. Their paper appeared in the same issue of Nature, alongside papers by Wilkins and by Franklin, presenting their X-ray evidence. But it was Watson and Crick's model that everyone remembers. They had found the double helix. And here's where we need to pause and talk about Rosalind Franklin, because the way this story has been told for decades is not entirely fair to her, and the way her contribution has been recognized has been one of the great ongoing reckonings of the scientific community. Franklin's data, particularly Photo 51, was essential to Watson and Crick's discovery. Without it, they could not have built their model when they did. But Franklin's work was shared with Watson without her consent. She was not told that her data was being used. She was not consulted. She was not credited as a full partner in the discovery, at least not at the time. Watson's later memoir, The Double Helix, published in 1968, portrayed Franklin in a dismissive and sometimes cruel way, focusing on her appearance and temperament rather than her scientific brilliance. The book was controversial from the moment it appeared, and Crick and Wilkins both objected to Watson's characterization of events. Franklin herself never had the chance to respond. She died in 1958, at the age of 37, of ovarian cancer, possibly related to her extensive exposure to X-rays in the course of her work. She died without knowing the full significance of the controversy that would later surround her contribution. In 1962, Watson, Crick, and Wilkins shared the Nobel Prize in Physiology or Medicine for the discovery of the structure of DNA. The Nobel Prize is not awarded posthumously, so Franklin was not eligible. Whether she would have been included had she lived is a question that the scientific community has debated ever since, and the consensus now is that she absolutely should have been. Her experimental work was as important as anyone's in the story, and the fact that it was used without her knowledge makes the omission even more painful. In recent years, Franklin has been increasingly recognized for her contributions. In 2023, a study of her lab notebooks confirmed that she had independently deduced many of the structural features of DNA before Watson and Crick built their model. She was closer to the answer than she's traditionally been given credit for. She was not, as Watson's memoir implied, someone who had the data but couldn't see what it meant. She was a brilliant scientist working systematically toward the truth, whose work was taken and used by others who got there first by a different method. So that's the story of the double helix. It's a story of scientific triumph and ethical failure, of brilliant deduction and casual injustice, and it ended with the revelation of a structure so elegant that it almost seems designed. Two strands, wound together, each the mirror image of the other, carrying within their sequence of bases all the information needed to build and maintain a living organism. The secret of life, coiled up in every cell. But discovering the structure was just the beginning. Knowing that DNA is a double helix of paired bases tells you how it stores information and how it copies itself, but it doesn't tell you how that information is actually used. How does a sequence of A's and T's and G's and C's actually build a protein? How does a gene become an eye color, a blood type, an enzyme that digests your dinner? The structure of DNA was solved in 1953. The code, the actual language in which genetic instructions are written, would take another decade to crack. And the story of how that happened involves a whole new cast of characters, a worldwide race to decipher nature's most fundamental alphabet, and a moment in 1961 when a relatively unknown scientist stood up at a conference in Moscow and announced results so stunning that the audience erupted in applause that went on for several minutes. Let's get there. So by 1953, you know the shape of DNA. a double helix, 4 bases, adenine, thymine, guanine, cytosine, paired up in the middle like the rungs of a twisted ladder. And you know that the sequence of those bases must somehow encode genetic information, because what else could it be? The sugar-phosphate backbone is the same everywhere, the same repeating pattern, so it can't carry information any more than the binding of a book tells you what's written on the pages. The information has to be in the sequence of bases, the order of A's, T's, G's, and C's along the strand. That's the text. The question is, what language is it written in? Now, here's the basic problem. DNA has 4 letters, A, T, G, C. Proteins, which are the workhorses of the cell, the molecules that actually do most of the work of being alive, are made of 20 different amino acids. So somehow, a 4-letter alphabet has to specify a 20-word vocabulary. How? Well, think about it mathematically. If each amino acid were specified by a single base, you'd only have 4 options. That's not enough. If each amino acid were specified by a pair of bases, you'd have 4 times 4, which is 16 options. Still not enough. You need at least 20. But if each amino acid is specified by a triplet of bases, 3 bases in a row, you get 4 times 4 times 4, which is 64 options. That's more than enough. You've got 64 possible triplets to code for 20 amino acids, with plenty of room left over for redundancy, which turns out to be exactly what nature does. Most amino acids are coded for by more than one triplet. The triplets are called codons, and the mapping from codons to amino acids is called the genetic code. This reasoning, that the code must be a triplet code, was first proposed by the physicist George Gamow in 1954. Gamow was a character, a Russian-born theoretical physicist who had made major contributions to cosmology and nuclear physics. He was one of the key figures behind the Big Bang theory, and who dabbled in biology with infectious enthusiasm. He read Watson and Crick's paper and immediately fired off a letter suggesting that the code must work in groups of 3. He even founded an informal club, the RNA Tie Club, with 20 members, 1 for each amino acid, who exchanged ideas about the code by mail. Each member got a custom-made tie with an embroidered RNA motif. Crick and Watson were members. So was Richard Feynman. It was that kind of era, playful, freewheeling, people from different fields crashing into each other's problems with fresh eyes. But figuring out that the code was probably a triplet code was one thing. Actually deciphering it, figuring out which triplet corresponds to which amino acid, was something else entirely. And that challenge would take nearly a decade and involve some of the most ingenious experiments in molecular biology. But before we get to the code breaking, there's a crucial intermediate step that needs explaining, because it tells you how the cell actually reads the DNA. And this is where things get really elegant. See, DNA lives in the nucleus of the cell, locked away in the command center. But proteins are built outside the nucleus, in the main body of the cell, on molecular machines called ribosomes. So the DNA can't directly supervise protein construction. It's like having the master blueprint locked in a vault. You need to make a working copy that can be carried out to the construction site. That working copy is RNA, specifically messenger RNA or mRNA, and the process of making it is called transcription, which is a perfect metaphor. You're transcribing information from one form to another, like a scribe copying a manuscript. An enzyme called RNA polymerase reads along one strand of the DNA and builds a complementary strand of RNA. The RNA is almost identical to DNA. It uses the same bases, except that it has uracil instead of thymine, and its sugar backbone is slightly different. But functionally, it's a portable copy of the gene. The mRNA strand peels off, leaves the nucleus, and travels to a ribosome, where the actual protein construction happens. And here's the next beautiful piece. At the ribosome, the mRNA is read 3 bases at a time, 1 codon at a time, and for each codon, a specific amino acid is brought in and added to the growing protein chain. The amino acids are carried by yet another type of RNA, called transfer RNA, or tRNA. Each tRNA molecule has an anticodon on one end, 3 bases that match a specific codon, and the corresponding amino acid attached to the other end. It's like a molecular delivery service. The ribosome reads a codon. The matching tRNA shows up with its amino acid. The amino acid gets attached to the chain, and the ribosome moves on to the next codon. One by one, amino acid by amino acid, the protein is built according to the instructions written in the DNA. This whole process, DNA to RNA to protein, was described by Francis Crick in 1958 as the central dogma of molecular biology. Information flows from DNA to RNA to protein. That's the direction. DNA is the master copy. RNA is the messenger. Protein is the product. And while there are exceptions, we'd later discover that some viruses can go from RNA back to DNA, for instance. The central dogma captures the essential logic of how genetic information is used in virtually all living things. It's one of the most unifying principles in all of biology. Now, back to cracking the code. By the late 1950s, everyone knew the code was almost certainly a triplet code, and the general flow of information from DNA to RNA to protein was understood in outline. But the actual dictionary, which codon specifies which amino acid, was still unknown. And this is where Marshall Nierenberg enters the story. And honestly, his story is one of my favorites in all of science, because he was not someone anyone expected to crack one of the biggest puzzles in biology. Nierenberg was a young biochemist at the National Institutes of Health in Bethesda, Maryland. He was 34 years old, not at one of the famous universities, not part of the in-crowd of molecular biology, not a member of the RNA Tie Club. He was, by the standards of the field's elite, an outsider, and in 1961, he and his postdoctoral fellow, Heinrich Matthaei, performed an experiment so simple and so brilliant that it broke the code wide open. Here's what they did. They built a cell-free system, a test tube containing all the molecular machinery needed to make proteins, extracted from bacterial cells, ribosomes, tRNAs, amino acids, energy molecules, everything. Just add an mRNA template, and the system would obediently churn out proteins. So they made a synthetic mRNA that contained only one type of base, uracil, a long chain of nothing but U's. Poly-U, they called it. U, over and over. They added this to their cell-free system and waited to see what protein it would make. The answer was a chain of nothing but phenylalanine. One amino acid, repeated over and over. UUU codes for phenylalanine. They had deciphered the first word of the genetic code. The experiment was done on May 27, 1961. Matthaei actually ran the critical experiment at 3 in the morning because the system worked better at certain temperatures, and he wanted optimal conditions. Think about that. One of the most important experiments in the history of biology, performed in the dead of night by a postdoc who couldn't sleep. Nierenberg presented the results at the International Congress of Biochemistry in Moscow in August 1961. His first presentation was to a small session with only about 35 people in the audience. Nobody really noticed. But then, Matthew Messelson, who happened to attend that small session and grasped the significance immediately, arranged for Nirenberg to repeat the presentation in the larger auditorium to the full plenary session. Crick himself was chairing the session, and when Nirenberg finished, the room erupted. People realized that the code was being cracked, in real time, by someone nobody had heard of. It was one of those extraordinary moments in science when the hierarchy gets upended. Within a few years, using increasingly sophisticated versions of Nirenberg's approach and major contributions from Har Gobind Khorana, who synthesized RNA molecules with defined repeating sequences, and Robert Hawley, who determined the structure of tRNA, the entire genetic code was deciphered. All 64 codons were mapped to their corresponding amino acids or to stop signals. 3 of the 64 codons don't code for any amino acid. They're stop codons, the molecular equivalent of a period at the end of a sentence, telling the ribosome that the protein is complete. One codon, AUG, serves double duty. It codes for the amino acid methionine and also acts as the start signal, the capital letter at the beginning of every protein. And here's one of the most remarkable things about the genetic code. It's essentially universal. The same codons specify the same amino acids in bacteria, in plants, in fungi, in flies, in fish, in humans. There are a handful of minor variations in certain organisms. Some mitochondria use slightly different codons, for instance. But the core code is the same everywhere. Every living thing on Earth, from the bacteria in your gut to the oak tree outside your window to the whale in the deep ocean, reads the same genetic language. UU means phenylalanine everywhere. GCG means alanine everywhere. The code was established very early in the history of life, probably within the first billion years of Earth's existence, and it has been conserved, essentially unchanged, for roughly 4 billion years. That's a testament to how fundamental it is. Evolution can reshape bodies, create and destroy species, build brains and wings and eyes, but it doesn't mess with the code itself. The code is foundational. It's the operating system that everything else runs on. Nirenberg, Khorana, and Hawley shared the Nobel Prize in Physiology or Medicine in 1968. Nirenberg was 41 years old. The outsider had become one of the most celebrated scientists of his generation, so by the late 1960s, the picture was remarkably complete. DNA carries the genetic information in the sequence of its bases. The information is organized into genes, each gene being a specific stretch of DNA that codes for a specific protein. The information flows from DNA to mRNA through transcription, and from mRNA to protein through translation. The genetic code is a triplet code, with 64 codons mapping to 20 amino acids plus stop signals. And the code is universal. It's worth pausing to appreciate how elegant this system is. Think about it. The entire complexity of life, every organism you've ever seen or imagined, from a bacterium to a blue whale, is built using a language with only 4 letters, reading them 3 at a time, to make chains from an alphabet of 20 amino acids, which fold into proteins that do everything from catalyzing chemical reactions to building structural frameworks to carrying oxygen in your blood. The complexity doesn't come from complicated components. It comes from the combinatorial possibilities. 20 amino acids, arranged in chains of hundreds or thousands of units, folded into 3-dimensional shapes, determined by the sequence. The number of possible proteins is, for all practical purposes, infinite. It's like having 26 letters in an alphabet. Each individual letter is simple, but you can write Shakespeare with them. And now, with this understanding in hand, the structure, the code, the mechanism, biologists in the 1970s started asking a question that would have seemed like science fiction just a decade earlier. If DNA is a language, and we can read it, then can we also write it? Can we edit it? Can we cut a gene out of one organism and paste it into another? Can we rewrite the instructions of life itself? The answer, it turned out, was yes. And the tools that made it possible came from an unexpected source, from bacteria fighting for their own survival against viruses. Because bacteria, it turns out, have been doing genetic engineering for billions of years. They have molecular scissors that can cut DNA at specific sequences, and molecular glue that can paste it back together. And in the early 1970s, scientists figured out how to borrow those tools and use them for purposes that nature had never intended. The age of genetic engineering was about to begin, and it would start with a discovery about how bacteria defend themselves against the very same phages that Hershey and Chase had studied 20 years earlier, those viruses that inject their DNA into bacterial cells. Because here's the thing that nobody expected. Bacteria don't just sit there and take it. They fight back. And the weapons they use to fight back would become the most powerful tools in the history of biology. Let's talk about restriction enzymes. In the 1960s and early 1970s, microbiologists were studying a strange phenomenon. When certain viruses infected certain bacteria, the bacteria would sometimes survive by chopping up the invading viral DNA. The bacteria had enzymes, molecular scissors, that could recognize specific short sequences of DNA and cut both strands at that point. These enzymes were called restriction enzymes because they restricted the growth of viruses. And the reason the bacteria didn't chop up their own DNA was that they chemically modified their own DNA at those same sequences, adding little methyl groups that said, essentially, "This is ours, don't cut it." It was an immune system, billions of years old, elegant in its simplicity. The key insight came from Hamilton Smith at Johns Hopkins University, who in 1970 isolated a restriction enzyme from the bacterium Haemophilus influenzae and showed that it cut DNA at a specific, predictable sequence. Not at random, at a particular sequence of bases. Every time the enzyme encountered that sequence, it cut. Every time. That was huge because it meant you could use these enzymes as precision tools. If you knew the sequence, you could predict exactly where the enzyme would cut. You could take a long piece of DNA and chop it into defined, reproducible fragments. And then, in 1971 and 1972, several groups, most notably Daniel Nathans at Johns Hopkins and Paul Berg at Stanford, started using these enzymes to do something unprecedented. They began cutting and pasting DNA from different organisms. Berg, who is sometimes called the father of genetic engineering, used restriction enzymes to cut DNA from a monkey virus called SV40 and to splice it together with DNA from a bacteriophage, a completely different organism. He created the first recombinant DNA molecule, a chimera, a piece of genetic material that had never existed in nature, assembled from parts of two different viruses. It was a landmark moment, and Berg would share the Nobel Prize for it in 1980. But here's the thing that really set the revolution in motion. In 1973, Stanley Cohen at Stanford and Herbert Boyer at the University of California, San Francisco, took recombinant DNA to the next level. They figured out how to insert a foreign gene into a bacterial plasmid, a small circular piece of DNA that bacteria carry in addition to their main chromosome, and then get that plasmid back into a living bacterium, where it would replicate along with the bacterium's own DNA. They had created a system for cloning genes. You could take a gene from any organism, splice it into a plasmid, transform bacteria with it, and then grow billions of copies of those bacteria, each one carrying and replicating your gene of interest. You had a living factory, producing as many copies of any gene as you wanted. The first practical demonstration was electrifying. Cohen and Boyer inserted a gene from a frog, a gene for ribosomal RNA, into bacteria, and the bacteria dutifully replicated it, an amphibian gene running in a bacterial cell. The species barrier, the wall that had separated the genetic material of different organisms for billions of years, had been breached. And almost immediately, people started worrying, because the same technology that could clone frog genes in bacteria could, in theory, be used to clone anything. What if someone cloned a dangerous gene, a toxin, a drug resistance factor, and it escaped from the laboratory? What if recombinant bacteria got into the environment and caused unpredictable ecological damage? The potential risks were speculative, but they were not silly. You were creating organisms that had never existed, inserting genes across kingdom boundaries, and nobody had any experience with what might happen. The scientific community, to its credit, took these concerns seriously. In an almost unprecedented act of self-regulation, a group of leading scientists, organized largely by Paul Berg, called for a voluntary moratorium on certain types of recombinant DNA experiments until the risks could be assessed. And in February 1975, they convened a historic conference at the Asilomar Conference Center on the Monterey Peninsula in California. About 140 scientists from around the world gathered at this beautiful coastal retreat center to argue about safety guidelines for genetic engineering. The Asilomar Conference was tense, contentious, and ultimately productive. Scientists debated for days about what kinds of experiments should be restricted, what kinds of physical and biological containment measures should be required, and how to balance the extraordinary scientific potential of recombinant DNA with the genuine uncertainty about its risks. In the end, they agreed on a set of guidelines that allowed research to continue under strict safety protocols, with the level of containment increasing with the assessed risk of the experiment. It wasn't perfect, and plenty of people on both sides thought the guidelines were either too restrictive or not restrictive enough. But the Asilomar Conference established a principle that has echoed through every subsequent debate about emerging biotechnology: that scientists have a responsibility to think about the potential consequences of their work and to act accordingly, even if it means slowing down. And genetic engineering did not slow down. With the safety framework in place, the technology exploded. One of the first major practical applications was the production of human insulin. Before recombinant DNA technology, insulin for diabetics came from the pancreases of pigs and cows collected from slaughterhouses. Animal insulin works in humans, but it's not identical to human insulin, and some patients had allergic reactions. In 1978, scientists at the biotech startup Genentech, co-founded by Herbert Boyer, the same Boyer from the Cohen-Boyer cloning experiments, synthesized the gene for human insulin and inserted it into bacteria. The bacteria produced genuine human insulin, identical to the molecule your own pancreas makes. By 1982, recombinant human insulin was approved by the FDA and on the market. It was the first product of genetic engineering to reach patients, and it transformed the treatment of diabetes worldwide. Genentech itself was a landmark. Founded in 1976, it was essentially the first biotechnology company, the first company to take the tools of molecular biology and turn them into commercial products. Boyer invested $500 of his own money. Within a few years, the company was worth billions. The biotech industry was born. But while the genetic engineering revolution was transforming medicine and industry, another line of research was developing that would prove equally transformative. And it started with a very different kind of question. Not how to manipulate genes, but how to read them, how to determine the exact sequence of bases in a piece of DNA. Because if DNA is a book, the techniques developed so far had let scientists pull out chapters and copy them, but nobody could actually read the text yet. The sequence of A's, T's, G's, and C's in a gene, in a chromosome, in an entire genome, that was still unknown. Two methods for DNA sequencing were developed independently in the mid-1970s. One was by Alan Maxam and Walter Gilbert at Harvard, using chemical methods to break DNA at specific bases. The other, and this is the one that would ultimately dominate, was by Frederick Sanger at the Medical Research Council Laboratory of Molecular Biology in Cambridge, England. And Sanger is worth pausing on, because he was one of the most remarkable scientists of the 20th century, and also one of the most modest. Sanger was quiet, unpretentious, almost self-effacing. He described himself as just a chap who messes about in his lab. He had already won one Nobel Prize, in 1958, for determining the amino acid sequence of insulin, the first time anyone had worked out the complete chemical structure of a protein. That work alone would have made a career. But then he went on to develop a method for sequencing DNA that would change everything. Sanger's method, called chain termination sequencing or the dideoxy method, was elegant. It used modified versions of the 4 DNA bases, dideoxynucleotides, that could be incorporated into a growing DNA strand by DNA polymerase, but that, once incorporated, would terminate the chain. They were molecular stop signs. By running many copies of the reaction simultaneously, each one randomly terminated at a different position, you'd end up with a collection of fragments of every possible length. Separate those fragments by size using gel electrophoresis. Shorter fragments travel farther through the gel, and you could read the sequence directly from the pattern of bands. One band for each base, in order, from bottom to top. A, T, G, C, C, A, T, G. The text of life, readable at last. Sanger won his second Nobel Prize in 1980 for this work, making him one of only 4 people in history to win 2 Nobel Prizes. And characteristically, he downplayed it. He retired in 1983, saying he wanted to potter about in the garden. The institute where he worked was later renamed the Sanger Institute in his honor, and it would become one of the leading centers of the Human Genome Project. But before we get to the Genome Project, there's one more tool we need to talk about, because without it, modern genetics would be impossible. And it was invented in the most unlikely way imaginable, by a surfing, LSD experimenting, authority-defying biochemist who claimed the idea came to him while driving along a California highway on a Friday night in 1983. His name was Kary Mullis, and the tool was the polymerase chain reaction, or PCR. And PCR is, without exaggeration, one of the most important inventions in the history of biology. It's the reason we can do forensic DNA testing. It's the reason genome sequencing became practical. It's the reason COVID tests worked. It touches almost every area of modern biology, medicine, and forensics. Here's the problem PCR solves. In molecular biology, you constantly need to analyze specific sequences of DNA, but biological samples, a drop of blood, a swab from a crime scene, a fragment of ancient bone, contain only tiny amounts of DNA, mixed in with everything else in the cell. You need a way to take a specific stretch of DNA and make millions or billions of copies of it, amplify it, so you have enough to work with. Mullis's insight was that you could do this using DNA polymerase, the enzyme that copies DNA, in a cleverly designed cycle. Here's how it works. You heat up the DNA sample, which causes the 2 strands of the double helix to separate, to denature. Then you cool it down and add short synthetic pieces of DNA called primers. designed to bind to the sequences flanking the region you want to copy. DNA polymerase then extends from each primer, copying the template strand, creating new double-stranded DNA in the region between the primers. Now you have twice as many copies. Heat it up again, the strands separate. Cool it down, the primers bind, the polymerase extends. Now you have 4 copies. Heat, cool, copy. Heat, cool, copy. Each cycle doubles the number of copies. After 30 cycles, you've got over 1 billion copies. Starting from a single molecule of DNA, in a couple of hours, you have enough material to see on a gel, to sequence, to analyze. The key technical breakthrough that made PCR practical was the use of a heat-stable DNA polymerase. See, the heating step kills ordinary DNA polymerase. You'd have to add fresh enzyme after every cycle, which was tedious and expensive. But in the hot springs of Yellowstone National Park, there lives a bacterium called Thermus aquaticus, which thrives in boiling water. Its DNA polymerase, called Taq polymerase, works at high temperatures and isn't destroyed by the heating step. You add it once, and it survives through all the cycles. That little detail, an enzyme from a bacterium that lives in hot springs, turned PCR from a clever idea into a practical, automatable workhorse that transformed biology. Mullis won the Nobel Prize in Chemistry in 1993. He was a polarizing figure, brilliant, eccentric, and prone to controversial views on various scientific topics. But PCR was unquestionably a stroke of genius, and its impact has been immeasurable. And now, with DNA sequencing and PCR in the toolkit, the stage was set for the most ambitious project in the history of biology. A project that would attempt to read the entire genetic text of a human being, all 3 billion base pairs, every A, T, G, and C, on all 23 pairs of chromosomes. The Human Genome Project. It was proposed in the mid-1980s, formally launched in 1990, and it was, at the time, the largest coordinated biological research project ever undertaken. The initial goal was to complete the sequence by 2005, with a budget of $3 billion, roughly $1 per base pair. Many scientists thought it was impossible, or at the very least a colossal waste of money. The technology wasn't there yet, they argued. The genome was too big. The resources would be better spent on smaller, more focused research projects. They were wrong. The technology improved faster than anyone anticipated, driven partly by the pressure of the project itself. And the project got an unexpected jolt of competition in 1998, when a brash, controversial scientist named Craig Venter announced that his private company, Celera Genomics, would sequence the human genome faster and cheaper than the publicly funded project, using a different strategy called whole genome shotgun sequencing. Venter's approach was to skip the painstaking process of mapping genes to specific chromosomes, and instead just blast the entire genome into small fragments, sequence all of them, and use powerful computers to figure out how they overlapped and fit together. Many academics dismissed it as reckless. Some called it irresponsible. But it worked, and the competition between the public project, led by Francis Collins, and Celera, led by Venter, pushed both groups to accelerate dramatically. In June 2000, President Bill Clinton stood at a podium at the White House, flanked by Collins and Venter, and announced that the first draft of the human genome was complete. It was presented as a tie, a diplomatic fiction perhaps, since both groups had legitimate claims. The draft sequences were published simultaneously in February 2001, the public project in the journal Nature and Celera's version in Science. The finished sequence, covering about 99% of the gene-containing regions, was completed in 2003. 2 years ahead of the original schedule. And the genome was full of surprises. The biggest one was the gene count. Before the project, most estimates had put the number of human genes at somewhere between 50,000 and 100,000. After all, humans are complicated organisms. We must have a lot of genes, right? The actual number turned out to be far lower. The current best estimate is somewhere around 20,000 to 25,000 protein-coding genes. That's barely more than a roundworm, which has about 20,000. A fruit fly has about 14,000. A simple mustard plant has about 27,000, more than a human. The complexity of an organism, it turned out, doesn't come primarily from having more genes. It comes from how those genes are regulated, how they interact, how the same gene can produce different proteins through different processing of its RNA, and a host of other mechanisms that nobody fully appreciated before the genome was sequenced. And here's another surprise that turned the field upside down. Only about 1.5% of the human genome actually codes for proteins. 1.5%. The rest, the other 98.5%, was initially dismissed as junk DNA, a term that reflected the assumption that if a stretch of DNA didn't code for a protein, it must be useless debris, evolutionary leftovers, molecular fossils that had accumulated over billions of years of evolution but no longer served any purpose. That assumption, like the assumption that DNA was too boring to carry genetic information, turned out to be spectacularly wrong. And understanding why it was wrong, understanding what all that so-called junk DNA actually does, would open up entirely new fields of biology and completely reshape our understanding of how genes work. Because it turns out that the real story of genetics isn't just about genes. It's about everything around the genes: the switches, the dimmers, the regulators, the vast and intricate control network that determines when each gene is turned on, how loudly it's expressed, in which cells, at which times, in response to which signals. And that realization, that the genome is not just a parts list, but an exquisitely orchestrated regulatory system, that's where the story gets really interesting. Let's talk about gene regulation. Think about this. Every cell in your body, with a few exceptions, like red blood cells which lose their nucleus, carries the same DNA. The same complete set of instructions. The cells in your eyes have the same genome as the cells in your liver, which have the same genome as the cells in your skin, which have the same genome as the cells in your bones. And yet those cells look and behave completely differently. A nerve cell in your brain can be 3 feet long, with branching extensions that form thousands of connections. A red blood cell is a tiny, flexible disc packed with hemoglobin. A muscle cell is a long fiber that can contract. How can cells with identical DNA be so radically different? The answer is gene regulation. Not every gene is active in every cell. In a liver cell, the genes for liver-specific proteins are turned on, while the genes for, say, the light-sensitive proteins in your retina are turned off. In a skin cell, the genes for keratin and other structural proteins are active, while the genes for digestive enzymes are silent. The genome is like a massive cookbook with 20,000 recipes, and each cell type only opens to the pages it needs. The recipes that aren't being used are still there, still intact, but they're effectively closed, inaccessible, switched off. And the mechanisms that control this switching are astonishingly sophisticated. One of the earliest and most elegant was discovered by 2 French scientists, Jacques Monod and François Jacob, working at the Pasteur Institute in Paris in the late 1950s and early 1960s. They were studying a simple question: how does the bacterium E. coli know when to make the enzymes for digesting lactose, The sugar in milk? When there's no lactose around, the bacterium doesn't waste energy making those enzymes. But the moment lactose appears, the genes for the lactose-digesting enzymes switch on, and the bacterium starts producing them. How? Monod and Jacob figured out that there's a regulatory gene, they called it a repressor, that produces a protein which physically sits on the DNA near the lactose genes and blocks them. It's like putting your hand over a paragraph in a book so nobody can read it. When lactose shows up, it binds to the repressor protein and changes its shape, causing it to fall off the DNA. Now the genes are uncovered, the RNA polymerase can read them, and the enzymes get made. When the lactose is used up, the repressor goes back to its original shape, sits back down on the DNA, and the genes are silenced again. It's a feedback loop, a molecular switch, responsive and reversible. This was the operon model, and it won Jacob and Monod the Nobel Prize in 1965. It was the first clear demonstration that genes are actively regulated, that the cell is not just a machine that mindlessly reads its DNA from start to finish, but a sophisticated information processing system that makes decisions about which genes to use and when. Now, that's how it works in bacteria, which are relatively simple. In organisms like us, eukaryotes, cells with a nucleus, the regulatory machinery is far more complex, and this is where that so-called junk DNA becomes central to the story. It turns out that much of the non-coding DNA in the human genome is involved in gene regulation. There are enhancers, sequences of DNA, that can be located thousands or even millions of bases away from the gene they regulate, and that boost the gene's expression when specific proteins bind to them. There are silencers that do the opposite. There are insulators that prevent regulatory signals from leaking between adjacent genes. There are promoters, the sequences right at the beginning of a gene where RNA polymerase binds to start transcription. And there are transcription factors, proteins that bind to these regulatory sequences and either help or hinder the reading of the gene. A single gene might be regulated by dozens of different enhancers, each one active in a different tissue, at a different time, in response to a different signal. The combinatorial complexity is staggering. It's not just on or off. It's a dimmer switch with 100 different knobs, and the setting of each knob depends on what cell you're in, what stage of development you're at, what signals you're receiving from neighboring cells, what hormones are circulating in your blood, and even what you ate for breakfast. And this brings us to a field that has exploded in the last few decades and that has fundamentally changed how we think about inheritance. Epigenetics. The word literally means above genetics, and it refers to changes in gene expression that are heritable, that can be passed from cell to daughter cell, and in some cases from parent to offspring, without any change in the DNA sequence itself. The letters of the genetic text stay the same, but the way they're read changes. Think of it like this. If the DNA sequence is the text of a book, then epigenetics is the set of annotations, the highlights, the bookmarks, the sticky notes, the pages that have been glued shut. The text hasn't changed, but which parts you can access and how you interpret them has been profoundly altered. The most well-studied epigenetic mechanism is DNA methylation. Cells can attach small chemical groups, methyl groups, each just 1 carbon and 3 hydrogens, directly to certain bases in the DNA, usually to cytosine. When a gene's promoter region is heavily methylated, the gene is typically silenced. The methyl groups don't change the sequence. The C is still a C, but they effectively make that stretch of DNA harder to read. It's like smearing invisible ink over a paragraph. The words are still there, but the machinery can't access them easily. And here's what makes this epigenetic. When a cell divides, The methylation pattern is usually copied along with the DNA. So if a gene is methylated and silenced in a liver cell, it stays silenced in the daughter liver cells. The identity of the cell, what type it is, what genes it expresses, is maintained across generations of cell division, even though every cell has the same underlying DNA. Another major epigenetic mechanism involves histone modifications. Remember, in the nucleus of your cells, DNA isn't just floating around loose. It's wrapped around small proteins called histones, like thread around tiny spools. And the way the DNA is wrapped, tightly or loosely, affects whether the genes in that region can be read. Chemical modifications to the histone proteins, adding acetyl groups, methyl groups, phosphate groups, and others, can loosen or tighten the wrapping, opening or closing access to the genes underneath. Loosely wrapped DNA, called euchromatin, is accessible and its genes can be active. Tightly packed DNA, called heterochromatin, is condensed and its genes are typically silent. The discovery that these epigenetic marks can sometimes be influenced by environmental factors was a bombshell. It means that your experiences, what you eat, what toxins you're exposed to, what stresses you endure, can, in some cases, alter the way your genes are expressed, not by changing the DNA itself, but by changing the annotations on it. And some of these changes can potentially be passed to your children. The most famous example comes from a terrible natural experiment, the Dutch hunger winter. In the winter of 1944-45, near the end of World War II, a German blockade cut off food supplies to the Western Netherlands. About 4.5 million people were affected by severe famine. Pregnant women were among those who suffered, and their children, born during or just after the famine, showed increased rates of obesity, cardiovascular disease, and other health problems decades later. That wasn't entirely surprising. Fetal malnutrition can have lasting effects. But what was surprising was that some studies suggested that the grandchildren of women who were pregnant during the famine also showed effects. The experience of starvation seemed to have left an epigenetic mark that could be transmitted across generations. Now, this area of research is still controversial. The evidence for robust transgenerational epigenetic inheritance in humans is debated, and some of the early studies have been challenged on methodological grounds. The effects are clearer in plants and in some animal models. But the basic principle that environmental factors can modify gene expression through epigenetic mechanisms, and that some of these modifications can persist, is well established. It adds a whole new layer of complexity to the story of genetics. Mendel's laws still hold. DNA is still the carrier of genetic information, but the way that information is read and expressed is far more fluid, far more responsive to the environment than anyone imagined when the double helix was first revealed. And here's where the story takes its most dramatic modern turn. Because in the early 2010s, a discovery emerged from an unlikely corner of microbiology, from the study of how bacteria defend themselves against viruses, that would give scientists a tool so powerful, so precise, so easy to use, that it would transform biology almost overnight. And that tool, which you've almost certainly heard of, is called CRISPR. CRISPR, clustered, regularly interspaced, short, palindromic repeats. Even the name is unwieldy, but the technology behind it is, in a word, revolutionary. And the story of how it was discovered is one of those beautiful threads in science, where decades of obscure, seemingly impractical research suddenly converge into something world-changing. The story starts in 1987, when a Japanese molecular biologist named Yoshizumi Ishino, working at Osaka University, noticed something odd in the genome of E. coli. There were these strange repetitive sequences in the DNA, short palindromic sequences, repeated over and over, separated by unique spacer sequences. Nobody knew what they were for. They were catalogued and essentially forgotten. Then, in the early 2000s, several researchers independently figured out what the spacers were. They were fragments of viral DNA. Somehow, bacteria were capturing snippets of DNA from viruses that had attacked them and incorporating those snippets into their own genome between the repeats. It was an archive, a molecular memory of past infections. And when the same virus attacked again, the bacterium could use that archived sequence to recognize the invader and destroy it. The mechanism, worked out over the next decade by many groups, was stunning. The bacterium transcribes the spacer sequence into a short piece of RNA. That RNA, called a guide RNA, pairs up with a protein called Cas9, a molecular scissor. The guide RNA leads Cas9 to the matching viral DNA, and Cas9 cuts it. Snip. The viral DNA is destroyed. The infection is stopped. It's an adaptive immune system, billions of years old, found in roughly half of all bacteria and most archaea. And it works with remarkable precision. The guide RNA recognizes its target by base pairing, the same AT and GC matching that holds the double helix together. It finds the matching sequence in the viral genome and directs the cut with extraordinary accuracy. Now, here's where it goes from fascinating microbiology to world-changing technology. In 2012, two scientists, Jennifer Doudna at the University of California, Berkeley, and Emmanuel Charpentier, then at Umeå University in Sweden, published a landmark paper demonstrating that the CRISPR-Cas9 system could be programmed. You could design a synthetic guide RNA to match any DNA sequence you wanted, pair it with Cas9, and the system would cut the DNA at precisely that location. Any gene, in any organism. You just had to change the sequence of the guide RNA. It was like having a word processor's find and replace function for DNA. Find this sequence, cut here. And once you've cut the DNA, the cell's own repair machinery kicks in. If you just let it repair naturally, the repair is often imperfect. It introduces small errors that disrupt the gene, effectively knocking it out. But if you provide a template, a piece of DNA with the sequence you want, the cell will sometimes use that template to repair the cut, inserting your desired sequence in place of the original. You can delete genes, insert new ones, correct mutations, change single letters of the genetic code, with a specificity and ease that would have seemed like magic to the scientists of even a decade earlier. The impact was immediate and explosive. Within months of the 2012 paper, labs around the world were using CRISPR to edit genes in bacteria, yeast, plants, flies, fish, mice, and human cells in culture. The technology was cheap, a few $100s for the reagents. It was fast. Experiments that would have taken months with older genetic engineering techniques could be done in weeks. And it was accessible. Any lab with basic molecular biology equipment could do it. CRISPR democratized genetic engineering in a way that nothing else had. Doudna and Charpentier were awarded the Nobel Prize in Chemistry in 2020, just 8 years after their landmark paper, which is extraordinarily fast for a Nobel. They were the first two women to share a Nobel Prize in science without a male co-laureate, and their discovery had already spawned hundreds of potential applications in medicine, agriculture, and basic research. But CRISPR also raised profound ethical questions. Questions that came to a dramatic head in November 2018, when a Chinese biophysicist named He Jiankui announced that he had done something that no one had done before, and that most scientists thought no one should do. He had used CRISPR to edit the genomes of human embryos, embryos that were then implanted, carried to term, and born. Twin girls, known by the pseudonyms Lulu and Nana. He claimed to have disabled a gene called CCR5, which the HIV virus uses to enter cells, with the goal of making the girls resistant to HIV infection. The reaction from the scientific community was swift, nearly unanimous, and overwhelmingly negative. What He Jiankui had done was widely condemned as reckless, unethical, and scientifically unjustified. The girl's father was HIV positive, but there were established, safe methods for preventing HIV transmission from parent to child that didn't require editing the embryo's DNA. The CCR5 edit was not well characterized. There were concerns about off-target effects, unintended cuts elsewhere in the genome. The gene he targeted, CCR5, has other functions in the immune system, and disabling it might have unforeseen health consequences. And most fundamentally, editing the genome of embryos that will become living people means those edits will be passed on to all of their descendants, forever. You're making a change to the human germline, to the genetic heritage of the species, without knowing the long-term consequences. He Jiankui was widely denounced. He was fired from his university, convicted by a Chinese court in December 2019, and sentenced to 3 years in prison. The affair sent shockwaves through the scientific community and led to renewed calls for international regulation of human germline editing. In 2020, an international commission convened by the U.S. National Academies and the Royal Society concluded that heritable human genome editing was not yet safe or effective enough to be used clinically, and that extensive public discussion and robust governance frameworks would be needed before it could ever be considered. But the CRISPR babies also crystallized a deeper question that had been simmering since the earliest days of genetic engineering. Where is the line? The technology to edit the human genome exists. It's getting better every year. The potential benefits, curing genetic diseases, eliminating heritable conditions that cause immense suffering, are real and compelling. But the potential for misuse, enhancement, discrimination, unforeseen consequences is equally real. And the decisions we make about this technology in the next few decades will shape the human species and human society in ways we're only beginning to imagine. And that's a big thing to sit with. But let's leave it for a moment and come back to something a bit more grounding. Because while the ethical debates are important, they sometimes overshadow the incredible things that genetics is actually doing right now, today, to help people. Real therapies, real cures, real lives being changed by our understanding of DNA. And the story of how gene therapy went from a disastrous early failure to one of the most promising frontiers in medicine is a story of persistence, humility, and the long, hard work of getting things right. Let's talk about gene therapy. The idea is almost absurdly simple. If a disease is caused by a faulty gene, a gene that's missing or mutated or not working properly, then why not just fix the gene? Give the patient a working copy. Replace the broken instruction with a correct one. In principle, it's the most logical approach to genetic disease you could imagine. In practice, it turned out to be extraordinarily difficult, and the early history of gene therapy includes one of the most painful chapters in modern medicine. The concept of gene therapy has been around since the 1960s, almost as soon as scientists understood what genes were and how they worked. But the first real attempt at human gene therapy came in 1990, when a team led by W. French Anderson at the National Institutes of Health treated a 4-year-old girl named Ashanti da Silva. Ashanti had a rare genetic disorder called severe combined immunodeficiency, or SID, specifically, a form caused by a defect in the gene for the enzyme adenosine deaminase, or ADA. Without this enzyme, Her immune system couldn't function. She was vulnerable to infections that a healthy child's body would fight off effortlessly. Children with this condition were sometimes called bubble babies because some had to live in sterile plastic enclosures to avoid deadly infections. The team took white blood cells from Ashanti, used a retrovirus, a virus that inserts its own genetic material into the host cell's DNA, to carry a working copy of the ADA gene into those cells, and then infused the corrected cells back into her body. It worked, partially. Ashanti's immune function improved. She was able to go to school, live a relatively normal life. The treatment didn't cure her completely. She still needed supplemental enzyme injections, but it was a proof of concept. Gene therapy could work in a human being. The field surged with optimism. Dozens of clinical trials were launched in the 1990s, targeting various genetic diseases. And then, in 1999, something terrible happened. Jesse Gelsinger was 18 years old. He had a mild form of a metabolic disorder called ornithine transcarbamylase deficiency, or OTC deficiency. His condition was manageable with diet and medication. He wasn't critically ill. He volunteered for a gene therapy trial at the University of Pennsylvania, led by James Wilson, not because he needed the treatment for himself, but because he hoped it would help others with more severe forms of the disease, including babies who died from it in infancy. He wanted to help. On September 13, 1999, doctors injected a massive dose of adenovirus, a common cold virus, modified to carry a working copy of the OTC gene, directly into Jesse's liver, through a catheter in his hepatic artery. Within hours, Jesse developed a severe immune reaction. His body attacked the viral vector with overwhelming force. His temperature spiked. His blood began to clot uncontrollably. His organs failed one by one. 4 days later, on September 17th, Jesse Gelsinger died. The death of Jesse Gelsinger devastated the gene therapy field. Investigations revealed serious problems with the trial. There were questions about whether the risks had been adequately disclosed to Jesse and his family, about whether the trial should have been halted earlier when other patients showed adverse reactions, about conflicts of interest involving the lead researcher. The FDA shut down gene therapy trials across the country. Funding dried up. Careers were ruined. The whole enterprise came to be viewed, fairly or not, as reckless. A field that had been so dazzled by its own promise that it had outrun its safety data. And then, in 2000, just months after Jesse's death, a gene therapy trial in France offered both hope and another devastating setback. Alain Fischer and Marina Cavazzana at the Necker Hospital in Paris were treating children with X-linked SCID, another form of the severe immunodeficiency that had afflicted Ashanti da Silva, but caused by a different gene defect. They used a retroviral vector to insert a working copy of the gene into the children's bone marrow stem cells, and it worked beautifully. The children's immune systems were restored. They went home, went to school, lived normal lives. It was hailed as the first unambiguous cure by gene therapy. But then, starting in 2002, several of the treated children developed leukemia. The retroviral vector had inserted the therapeutic gene near an oncogene, a gene that, when activated inappropriately, can trigger cancer. The very act of fixing one genetic problem had inadvertently created another. One child died. The trial was halted, modified, restarted with improved vectors, and eventually resumed with better safety records. But the damage to the field's reputation was profound. It took more than a decade for gene therapy to recover. What saved it was better vectors, better delivery vehicles. Scientists developed new types of viral vectors, particularly adeno-associated viruses, or AAVs, which are small, non-pathogenic viruses that can deliver genes into cells without integrating randomly into the chromosome, greatly reducing the risk of accidentally activating oncogenes. They learned to use lower doses, to choose target cells more carefully, to design clinical trials with more rigorous safety monitoring. And slowly, carefully, gene therapy started working. In 2017, the FDA approved the first gene therapy for an inherited disease in the United States, Luxturna, developed by Spark Therapeutics, for a form of inherited blindness called Leber congenital amaurosis. Patients who had been legally blind could see after a single injection of a working copy of the RPE65 gene directly into the retina. Children who had never seen stars could look up at the night sky. Think about that for a moment. A child who has spent their entire life in darkness, whose retinal cells have never functioned properly because of one misspelled gene, and a doctor injects a tiny amount of fluid into their eye, and the fluid contains viruses carrying the correct gene, and those viruses infect the retinal cells and deliver the working instruction, and the cells start producing the missing protein, and light becomes meaningful, and the child can see. It's hard to overstate how extraordinary that is. Since Luxturna, the pace has accelerated. In 2019, the FDA approved Zolgensma for spinal muscular atrophy, a devastating genetic disease that is the leading genetic cause of death in infants. Zolgensma delivers a working copy of the SMN1 gene using an AAV vector in a single intravenous infusion. Babies who would have been unable to sit, stand, or breathe on their own are now meeting developmental milestones. The drug costs about $2 million per dose, making it one of the most expensive medications in history, but it's a one-time treatment that can save a life. In December 2023, the FDA approved 2 gene-based therapies for sickle cell disease, Casgevy, which uses CRISPR to edit the patient's own bone marrow stem cells, and Lyfgenia, which uses a lentiviral vector. Sickle cell disease affects millions of people worldwide, predominantly people of African descent, and it causes chronic pain, organ damage, and shortened lifespan. These new therapies offer the possibility of a functional cure. And this is perhaps the most remarkable thing about the CRISPR-based therapy for sickle cell disease. It brings the story full circle in a way that's almost poetic. The disease is caused by a single base pair change in the gene for beta-globin, one of the components of hemoglobin. Just one letter of the genetic code out of 3 billion is wrong. A in the normal gene, T in the sickle cell gene. That single change causes the hemoglobin protein to fold slightly differently, which causes red blood cells to deform into a sickle shape, which causes them to clog blood vessels, which causes pain and organ damage. One letter, one changed amino acid, one altered protein, a lifetime of suffering. And now, CRISPR can go in and fix it. Not by correcting that single base. The current approved therapy actually takes a slightly different approach, reactivating a fetal form of hemoglobin that compensates for the defective adult form, but the principle is the same. We can read the error in the code, and we can intervene. We've gone from Mendel counting peas in a monastery garden to editing the spelling of the human genome, one letter at a time, in about 150 years. Let's shift gears a bit, because there's another branch of this story that deserves attention, and it's one that you interact with more directly than you might realize. DNA forensics. The use of genetic analysis to identify individuals, solve crimes, exonerate the innocent, and rewrite history. The idea of using DNA to identify individuals was pioneered by a British geneticist named Alec Jeffreys at the University of Leicester. In September 1984, Jeffreys was studying the structure of genes when he noticed something. When he looked at X-ray images of DNA from different members of the same family, each person's pattern was unique. The patterns were similar between relatives, more similar than between strangers, but no two people had the same pattern, not even close. Jeffries immediately realized the implications. He later described the moment as a eureka experience, saying he thought, "My God, what have we stumbled upon here?" What he'd stumbled upon was the fact that certain regions of human DNA are highly variable between individuals. These regions, called variable number tandem repeats, or VNTRs, consist of short sequences repeated over and over, and the number of repeats varies enormously from person to person. By using restriction enzymes to cut the DNA and gel electrophoresis to separate the fragments by size, you could create a pattern of bands, a genetic fingerprint, that was essentially unique to each individual. The probability that two unrelated people would have the same DNA fingerprint was astronomically low, 1 in billions or better. The first forensic use of DNA fingerprinting came in 1986, in a case in Leicestershire, England, right in Jeffries's backyard. Two teenage girls had been murdered in separate incidents in 1983 and 1986, and a local man had confessed to one of the killings. But when Jeffries tested the suspect's DNA against samples from the crime scenes, the results were clear. The suspect's DNA didn't match either crime scene. He was innocent. His confession was false. He became the first person in history to be exonerated by DNA evidence. The police then undertook an extraordinary step. They asked every adult male in the local area, over 5,000 men, to voluntarily provide a blood sample for DNA testing. After months of testing, the real killer was caught when a man named Ian Kelly was overheard in a pub saying he'd been paid to submit a blood sample on behalf of someone else. That someone else was Colin Pitchfork, a local baker. Pitchfork's DNA matched both crime scenes. He was arrested, convicted, and sentenced to life in prison. It was the first murder conviction based on DNA evidence, and it launched a revolution in criminal justice. Since then, DNA forensics has transformed law enforcement. The FBI's Combined DNA Index System, CODIS, contains millions of DNA profiles. Cold cases that had gone unsolved for decades have been cracked when DNA evidence was retested with newer, more sensitive techniques. And the Innocence Project, founded in 1992 by Barry Scheck and Peter Neufeld, has used DNA testing to exonerate more than 375 people who were wrongly convicted of crimes in the United States, including 21 who had been sentenced to death, 21 people who were on death row for crimes they didn't commit, freed by DNA evidence. That's not an abstraction. Those are real lives, saved by our ability to read the genetic code. But DNA has also reshaped our understanding of history and human identity in ways that go far beyond forensics. Ancient DNA, extracted from bones and teeth preserved for thousands or even tens of thousands of years, has revolutionized archaeology and anthropology. Svante Pääbo, a Swedish geneticist at the Max Planck Institute for Evolutionary Anthropology in Leipzig, spent decades developing techniques for extracting and sequencing DNA from ancient specimens, work that culminated in the sequencing of the Neanderthal genome, published in 2010, and the discovery of the Denisovans, an entirely unknown group of archaic humans identified solely from DNA extracted from a tiny finger bone found in a cave in Siberia. Pääbo won the Nobel Prize in Physiology or Medicine in 2022 for this work. And what ancient DNA has told us is humbling and fascinating. Modern humans didn't simply replace Neanderthals and Denisovans. We interbred with them. If you're of European or Asian descent, roughly 1 to 4% of your DNA comes from Neanderthals. People of Melanesian descent carry additional DNA from Denisovans. These ancient encounters left a genetic legacy that affects us today. Some Neanderthal gene variants influence our immune systems, our skin and hair, even our susceptibility to certain diseases. You carry within you the genetic echoes of encounters between different human species that happened 50,000 years ago. That's written in your DNA, right now, in your cells, as you lie here. And then there's the story that might be the most transformative of all, the one that bridges genetics and medicine in a way that affects everyone alive today. And it started, of all places, with a pandemic. Because the COVID-19 vaccines, developed by Pfizer-BioNTech and Moderna, the vaccines that were administered to billions of people worldwide, were not traditional vaccines. They were mRNA vaccines, and they worked by exploiting the same molecular machinery we've been talking about all night, the central dogma, the flow of information from nucleic acid to protein. Here's the idea. Instead of injecting a weakened or killed virus, or a piece of viral protein, you inject a synthetic mRNA molecule, a messenger RNA carrying the instructions for making just one protein from the virus, the spike protein that sits on the surface of SARS-CoV-2. Your cells take up the mRNA, feed it to their ribosomes, the same ribosomes that read your own mRNA every day, and the ribosomes obediently produce the viral spike protein. Your immune system sees the spike protein, recognizes it as foreign, and mounts an immune response. Antibodies are made, memory cells are created. And now, if the real virus shows up, your immune system is ready. It recognizes the spike protein and attacks immediately. The beauty of the mRNA approach is speed. Once you know the genetic sequence of the virus, and the sequence of SARS-CoV-2 was published in January 2020, within days of the virus being identified, you can design an mRNA vaccine almost immediately. You don't need to grow the virus in eggs or cell culture. You don't need to inactivate it. You just synthesize the mRNA in the lab, wrap it in a tiny lipid nanoparticle to protect it, and help it enter cells, and you have a vaccine candidate. The Moderna vaccine, mRNA-1273, was designed within 2 days of receiving the viral sequence. 2 days. It then took about 11 months of clinical trials to confirm that it was safe and effective, and by December 2020, vaccines were being administered. Now, the mRNA vaccine technology didn't appear out of nowhere. It was built on decades of foundational research by scientists like Katalin Karikó, a Hungarian-born biochemist who spent most of her career at the University of Pennsylvania working on mRNA therapeutics, largely in obscurity, struggling to get funding, at one point being demoted by her university because her grant applications kept getting rejected. For years, the scientific establishment considered mRNA therapeutics a dead end. The problem was that synthetic mRNA triggered a strong inflammatory response in cells. The immune system recognized foreign RNA as a sign of viral infection and attacked it before it could do anything useful. Karikó and her collaborator Drew Weissman discovered in the mid-2000s that by modifying one of the nucleotides in the mRNA, substituting pseudouridine for uridine, they could dramatically reduce the inflammatory response. The cells accepted the modified mRNA without sounding the alarm, and the protein was produced efficiently. This was the breakthrough that made mRNA vaccines possible, and for years, almost nobody cared. Karikó was pushed out of her university position. She eventually left Penn and joined BioNTech, the German company that would later partner with Pfizer to produce one of the 2 main COVID mRNA vaccines. In 2023, Karikó and Weissman were awarded the Nobel Prize in Physiology or Medicine. It was a vindication, decades in the making, a reminder that the most important breakthroughs often come from researchers working at the margins, pursuing ideas that the mainstream considers impractical, sustained by nothing more than curiosity and stubbornness, and the conviction that they're onto something real. And mRNA technology goes far beyond COVID vaccines. Clinical trials are underway for mRNA vaccines against influenza, RSV, HIV, and various cancers. The idea of personalized cancer vaccines, mRNA molecules custom designed to match the specific mutations in an individual patient's tumor, training their immune system to attack their specific cancer, is in clinical trials right now. It's the convergence of genomics and immunology, and decades of fundamental research into how DNA and RNA work, all coming together in ways that would have been unimaginable even 20 years ago. So here you are, lying in the dark, and the story of DNA and genetics stretches out behind you like a river. It starts in a monastery garden with a monk counting peas. It flows through microscopes aimed at dividing cells, through milk bottles full of fruit flies, through X-ray photographs and model building sessions, and a pub in Cambridge where someone announced the secret of life. It runs through the code breakers who deciphered the language of the gene, through the engineers who learned to cut and paste and edit that language, through the vast international effort to read the entire human text. It passes through tragedies, Jesse Gelsinger, the CRISPR babies, the long struggle for recognition of Rosalind Franklin, and through triumphs, children who can see, babies saved from fatal diseases, a pandemic vaccine designed in 48 hours. And it arrives here, at this moment, when we can read and write and edit the language of life with a precision that Mendel could never have imagined, that Watson and Crick could never have predicted, that even the scientists of 20 years ago would have found astonishing. And the thing that strikes me most, thinking about all of this, is how much of the story is about people. Stubborn, brilliant, flawed, persistent, sometimes lucky, often overlooked people. Mendel, counting his peas in the garden for 8 years, publishing his results, and being ignored for 3 decades. Avery, spending a decade proving that DNA was the genetic material, and never receiving the Nobel Prize. Franklin, doing the experimental work that was essential to the double helix discovery, and dying at 37 without seeing the full recognition of her contribution. Nierenberg, the outsider who cracked the code at 3 in the morning. Kariko, demoted and defunded, working for decades on an idea that almost everyone thought was a dead end, and then watching that idea save millions of lives in a pandemic. Science is done by people. And genetic science, more than almost any other field, connects us to the deepest questions about what it means to be a person. What makes you, you? How much of who you are is written in your DNA? And how much comes from the life you've lived, the food you've eaten, the air you've breathed, the people who've loved you? The answer, as far as genetics can tell us, is that it's both, always both. Your genes are not your destiny. They're your starting material. They're the hand you're dealt. And like any hand, what matters is how you play it, and what happens at the table, and who's sitting next to you. Every cell in your body, right now, is reading its DNA. Genes are being transcribed into mRNA. Ribosomes are translating that mRNA into proteins. Enzymes are repairing the small damages that accumulate in your DNA every day. About 10,000 to 100,000 molecular lesions per cell per day, quietly fixed by a fleet of repair enzymes that you never think about. Your DNA is being copied in cells that are dividing, in your gut lining, in your bone marrow, in your skin. The code that was established 4 billion years ago, the same code that Nierenberg cracked with his poly-U experiment, is running in you right now, flawlessly, automatically, the way it has in every living thing since the first cells. And the double helix is there, in every nucleus, coiled and supercoiled, and wrapped around its histones, and packed into chromosomes, and carrying within its sequence the information that makes you a human being and not a fruit fly, that makes you this particular human being and not any other. 3 billion base pairs, 20,000 genes, and all that regulatory DNA around them, the enhancers and silencers and switches, all that so-called junk that turned out to be anything but junk, orchestrating the most complex performance in nature, the construction and maintenance of a living human body. It's humbling, really. You carry within you a molecule that has been copied with astonishing fidelity for 4 billion years. Your DNA connects you in an unbroken chain of replication to the very first cells that ever lived on this planet. Every copy was made from a previous copy, which was made from a previous copy, stretching back through deep time, through the dinosaurs and the first fish, and the earliest multicellular life, and the ancient bacteria that thrived in hot springs and shallow seas. You are the current custodian of a library that has been accumulating information since life began. So let that thought settle in. Let it sit quietly. The molecules in your cells know what to do. They've known for a very long time. And as the mechanisms of life continue their work inside you, unspooling DNA, reading genes, building proteins, repairing damage, maintaining the elaborate molecular symphony that keeps you alive, you can rest. The code is running. It doesn't need your attention. It doesn't need your effort. It just runs, the way it's always run, reliably, elegantly, carrying 4 billion years of accumulated wisdom in a molecule that fits inside a space too small to see. That's a good thought to fall asleep with. The deepest knowledge of life is already inside you, written in a language older than any civilization, older than any species, older than the mountains and the oceans, and it's working perfectly. Right now, tonight, as you drift off. Sleep well. Your DNA will take it from here.